Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami microbiologist. Today in this video lecture we are going to talk about MacCokey agar. Now what is MacCokey agar? In the previous videos lecture, if you guys remember, we talked about that what is culture media and what is nutrient agar. We talked these two video lecture. If you missed these two video lectures, so go back to my channel and watch this video. Now this is another video lecture which is related to the culture media, and in this video lecture we are going to talk about MacCokey agar. So let's start the video. What is MacCokey agar? Again, this is the culture media, which is specially the selective media. Why it is called selective media? Because in this media we can only grow the gram-negative bacteria. Moreover, we are also called it differential media. Now why we call it differential culture media? Because in this media we can grow the lactose fermented bacteria as well as the non-lactose gram-negative bacteria as well. What does lactose fermented and non-lactose fermented gram-negative bacteria means? All of you better know gram-negative bacteria. Now lactose fermented means that this media contain lactose which act as a carbohydrate. So when the bacteria utilizing the lactose, so it can produce lactic acid, that is actually the acid, and that produce a red color or pink red colonies into a media, so we called it lactose fermented bacteria. Okay, now non-lactose fermented bacteria means those bacteria which do not utilize the lactose. So when the lactose is not utilizing, so definitely the media color is changed, means yellowish, or the yellowish colony or pale yellow colony appears. That is actually the means the indication of non-lactose fermenting bacteria. So that's why we can say that MacCokey agar is actually the selective and differential media. The MacCokey agar was actually discovered by a scientist named Alfred Theodore MacCokey at 20th century, means 1900. Okay, then what are the uses of this MacCokey agar? Remember guys, just like nutrient agar, as we talked that it is used for non-fastidious means non-fastidious bacteria. So similarly, MacCokey agar is also used for the growth of non-fastidious gram-negative rod bacteria. Okay, means for that those bacteria which do not need a special type of the nutrients, like E. coli as well, we can say Pseudomonas as well. So in this case we have Enterobacteriaceae family like Klebsiella, Enterobacter as well. Okay, so these all are actually non-fastidious bacteria. Then we have second point which explain that this media is specially used for the lactose fermented, to identify lactose fermented bacteria and non-lactose fermenting bacteria as well. We can also use this type of media to isolate the bacteria which is actually present in the water, dairy product, urine and feces. Okay, now what are the composition of this media? So remember guys, MacCokey agar contain peptone, means 17 gram for 1 liter. It contain meat casein, which is 3 gram for 1 liter, lactose monohydrate 10 gram for 1 liter, bile salt 1.5 gram for 1 liter, NaCl 5 gram for 1 liter, neutral red 30 mg for 1 liter, crystal violet 1 mg for 1 liter, agar 15 gram for 1 liter, and similarly distilled water is 1 liter. So these are the ingredient. Let's suppose if the media bottle is not available and these ingredients are available, so mix all these ingredients in 1 liter and then heat mix it and then autoclave, so you will get MacCokey agar. If MacCokey agar bottle is available, so don't proceed this process. Okay? Now then, what are the principle of this media? Means the ingredient which we talked, how it can use, what are actually the function of each ingredient, what are the importance of this ingredient which is present in the MacCokey agar. Remember guys, MacCokey agar mostly contain the four most important ingredient, which we called it lactose, bile salt, crystal violet, and neutral red. Okay, which these ingredients actually make the media selective as well as differential. Okay, now the bile salt, which is bile salt, as well as the crystal violet, which is present in this media, is actually acting as a selective agent. Why? Because it can inhibit the gram-positive bacteria and allow the gram-negative fastidious, non-fastidious bacteria. Okay, then we have lactose. So lactose is actually acting as a carbohydrate source. Okay, so when the bacteria ferment the lactose, means use the lactose, so after that the pH indicator, which is actually neutral red, are dropped down, means it can fall down. Due to this way, it can, the bacteria can produce pink red colonies. Then we have what we have. Remember guys, this is lactose fermented colony, means red pink colonies. So when we have non-lactose, so colorless colony or pale yellow colony will be appear, which can indicate that lactose is not utilized by bacteria. Then another point says that this media, as we talked, that it contain peptone, meat, casein. All these are actually acting as a nutritional component, and due to this way it can provide growth to the bacteria, and in this way the bacteria can divide it and increase their population. Then we have NaCl. So NaCl is actually this make the cell competent. Okay, remember guys, when NaCl is present inside the media, so the cell are become competent, and due to this way it can uptake the ingredient which is present outside of the cell, means it can also release out the ingredient which is actually present inside the cell, so it can release out the ingredient which is not needed by the cell. So what actually going on, due to this NaCl, the cell can maintain the nutrients balance. Okay, the nutrients which is actually required by the cell, they will get, and the nutrients which is actually means the waste material which is actually present inside the cell, so they will release out. This is actually the function of the NaCl which is present in the media. And as we talked, that agar is mostly used as a solidifying agent. How we can prepare the MacCokey agar? So remember guys, as we talked in the previous video lecture about the preparation of nutrient agar, so similarly we can also prepare the means MacCokey agar as we prepare the nutrient agar. But actually for 1 gram we required what? We required 50 gram for 1 liter MacCokey agar powder. Okay, and if we have ingredient, so we do not need this. If we do not have the ingredient, bottle is available, so then we need 50 gram for 1 liter. Okay, so mix 50 gram MacCokey agar powder in 1 liter, then heat, mix it, means at 50 degrees Celsius. After that autoclave it, means 121 centigrade for 15 minutes. After that, pour this media, after autoclave, pour this media into petri plate at a temperature of 50 to 60 centigrade. This is how we can prepare and use this MacCokey agar media. Now remember guys, the MacCokey agar is also, we can also make the MacCokey agar broth, MacCokey broth as well, which is also used for the bacterial growth. But you didn't see the MacCokey broth. Okay, MacCokey broth is not used as much as we can use the MacCokey agar. But here I write the point because MacCokey broth is also, we can also make it for a few purposes. Okay, not for the purposes as we use the nutrient broth and tryptic soy broth as well. Then requirements of MacCokey agar. So the same requirements which we talked in the nutrient agar, in this case we also need the same requirement, means we required petri plate, we required Bunsen burner, means spirit lamp, autoclave, hot plate. Okay, so these things are actually required for the preparation of the MacCokey agar. So this is actually the MacCokey agar. As you can see, this is the MacCokey agar media. Then we have the bacterial means characteristics which is actually showing on MacCokey agar. So remember guys, Now if we are going to talk E. coli, so E. coli mostly produce flat, dry, pink, non-mucoid surrounding dark pink area colonies in MacCokey agar. Okay, which is actually what? Which is the lactose fermented. So let me show the diagram. As you can see, this is what, this is the MacCokey agar, means this is the E. coli. So E. coli, as you can see, pink colonies we can see. Moreover, it can also produce hazy margin, as you can see these pink, around pink, dark pink color as well. So this is actually an identification of E. coli bacteria on MacCokey agar. Then we have what? The second is Pseudomonas aeruginosa. So Pseudomonas aeruginosa produce colorless, flat, smooth colonies having 2 to 3 mm in diameter, and it can also have greenish to brownish pigment, which showing that it is what? It is the non-lactose fermenting bacteria. NLF means non-lactose fermenting bacteria. So let me show the diagram. As you can see, this is what? This is the Pseudomonas aeruginosa growth on MacCokey agar. So as you can see, pale yellow colonies means this is non-lactose fermented bacteria on MacCokey agar. Then third is Enterobacter. So Enterobacter is actually producing pink mucoid colonies, which shows that it is also what? Lactose fermented bacteria. Let me show. So as you can see, this is what? This is the colonies of Enterobacter. Remember guys, most of the students get confused in the E. coli as well as in the Enterobacter. Okay, E. coli, if you want to identify E. coli, so with E. coli you will see around the colony, you will see pinkish, means dark pink area. Okay, you can see the dark pink area around the colonies, which is actually the bile salt precipitation. So in this case, in case of the Enterobacter, there is no dark pink area producer. Okay, this is how we can differentiate Enterobacter and E. coli. Then we have fourth, Salmonella. So Salmonella is actually what? This is the non-lactose fermenter bacteria. Okay, there are some lactose fermenter, but mostly Salmonella species are the non-lactose fermented bacteria. They can produce colorless colonies which have 2 to 3 mm convex colony with serrated margins. Okay, now let me show the diagram. This one, as we can see, this is what? This is the Salmonella gallnerium. Okay, now this pinkish is actually Salmonella gallnerium, and this one, as you can see, these are the Salmonella typhorium. So Salmonella species, if we talk to, so we have Salmonella gallnerium and Salmonella typhorium. Now Salmonella gallnerium is actually showing pink color colonies, which is actually lactose fermented, and Salmonella typhorium is actually showing yellowish colonies, as you can see, which is actually the non-lactose fermenter bacteria. Then we have Klebsiella, which can produce large mucoid pink pigment colonies, which is lactose fermented bacteria. And this, as you can see, this fourth diagram, this is the lactose fermented bacteria, and this is how we can see this is the growth of Klebsiella on MacCokey agar. Remember guys, the colony morphology, if you see, so with all these colony of the bacteria, the Klebsiella bacterial colony is larger as compared to all these. Okay, and is also pink color colony as well. Then we have Abomoni, which can show calcareous complex colony, means it is also non-lactose fermented colonies. Now this one is what? This is actually Abomoni growth on MacCokey agar, which is actually just like a chocolate color, as we can say. Okay, then we have Yersinia species growth. So Yersinia species have a colorless colony, just like peach. So what we can say, it is also non-lactose fermented bacteria, and this is actually Yersinia species, as you can see. Okay, a little bit it is actually, if you see the peach color, so peach color is also just like that, so means non-lactose fermented bacteria. Then we have Shigella. So Shigella produce pale colorless colonies with judge edges, means it is also non-lactose fermented bacteria, and it have 1 to 2 mm diameter. So as you can see, this first picture is actually salmonella, means shigella species. Okay, so it is also just like means white, half white colonies or colorless colonies, means it is also the non-lactose fermented bacteria. Then we have proteus, which can produce pale colonies with swarming, means it can spread to all throughout the media, and it can produce foul smell. If you spell it, so you will feel foul smell. Okay, and it means that it is also what, non-lactose bacteria. So this is actually the non-lactose bacteria, and this is the proteus characteristics on MacCokey agar. And as bile salt and crystal violet is present, so no growth of gram-positive bacteria will be present in MacCokey agar. Okay, so this is all about the MacCokey agar. I hope you get idea about that. Thank you so much.